Hi everyone, welcome to iGEM's Genetically Modified Organisms and Gene Editing slideshow. Today we'll be talking about various facets of what GMOs are, their impacts on humanity, and basically how they came to be, the science behind all of it. So what are GMOs? We'll give you some general information on this. GMOs are genetically modified organisms. They have undergone some sort of non-natural modification in their genes that changes something about it, from its physical appearance to the rate at which it grows. Now, where are GMOs found? They're found in plants, animals, and microorganisms. In plants, crops are commonly genetically edited in their genes to increase production rates and prevent invasive species from interfering, as well as quality control over produce. This is for the sake of consistency as well, and this benefits many U.S. markets. Now, animals are usually genetically modified in order to transfer beneficial traits across generations, such as maybe a chicken has more meat or something, or more meat, or they can consume less food and grow more fat, things like that. And for example, an animal may be modified to increase disease resistance or enhance its growth hormones as well. Microorganisms are used in human pathology to treat sickle cell diseases and genetic syndromes. Gene therapy is a common way to treat diseases such as certain cancers, heart disease, diabetes, and hemophilia. 13.5% of crops around the world are genetically modified in all. Now, 75% of crops from only the United States are genetically modified. Now let's go into the impact on humanity and some controversy behind GMO. So why do people think GMOs are bad? They think that they can cause cancer and other diseases, which is not necessarily true in every GMO. However, some studies that have proven that certain GMOs in food products are known to cause specific side effects, such as hepatic, pancreatic, renal, or reproductive effects. Pros: contains more nutrients and output. Fewer pesticides used in GMOs because the plant itself is resistant. Disease immunity increases in animals in GMO, and human diseases such as cancer and heart diseases can be treated. Now, there are cons to this as well, as this may increase the resistance to antibiotics. Health complications caused, including reproductive effects, and ethical issues. Should we really increase growth rates for livestock? How does gene editing work, and some explanations. The process of gene editing. So first, scientists choose the gene they want to substitute and what they want to substitute it with. And then potential candidates are chosen and tested with the help of genetic screens through a process called mutagenesis. And in order to identify the function of a specific gene in mutagenesis, one way is to introduce general mutations into an organism and compare it to a controlled subject, in hopes of detecting a difference in properties. Mutants are introduced into the organism's genome by exposing it to a mutagen, which changes the DNA structure. Common mutagens include UV light and C3H8SO3. This is ethylmethane sulfonate. Now we can just take a look at this. So, gene isolation. Once the gene is chosen, it must be isolated. The cell that contains the gene is opened and the DNA is purified. Then scientists isolate the gene by constructing a DNA library of that cell tissue organism. This library should include a fragment of the gene intended to be isolated, and these are often created using plasmid vectors. What are plasmid vectors? So plasmids are circular, self-replicating double strands of DNA that can be found in bacteria, archaea, and eukaryotic organisms, plasmids store for their host organism, providing things like antibiotic resistance. Plasmid vectors are the vehicles or tools used to drive recombinant DNA into a host cell and are necessary in genetic cloning. In cloning, this process takes modified DNA and has it reproduced in cells, creating a group of cells with the same DNA instead, same DNA strand. Now, recombinant DNA are DNA molecules formed by artificial methods of genetic recombination, and this creates sequences otherwise not found in the genome. And gene replication. The gene is then combined with the terminator's ending and the promoter's starting region before being inserted into the target organism. This starts and ends the transcription. Finally, the genetic material is inserted into a host genome, ending this process. Transcription is the process of making an RNA copy of a gene sequence. Terminators are DNA sequences that mark the end of a gene during transcription, and promoters are DNA sequences that define where the transcription of the gene begins. Now let's, So, some fields of application. Research scientists can provide insight for medical purposes, curing disease, as long as medicine. Genetic manipulation of mice for cancer, etc. Agriculture, we talked about this earlier in the slides, pest resistance, new drugs, animal modification, environment pig. Industry, genetically modified bacteria for biofuels. So these are, Let's do a quiz. So give 3 reasons why organisms are genetically modified. I'll give you a few moments. So, they increase production rates of plants, they quality control for plants, prevent invasive species for plants, transfer beneficial traits for animals, and treat diseases in microorganisms, all of which we've talked about in the slides. One common mutagen is C3H8SO3, or ethyl methane sulfonate. What is the other mutagen named earlier in this slideshow? I'll give you a little less time, but so this is UV light. We've talked about this. What shape are plasmids in comparison with other DNA strands? And a bonus, in which organisms are plasmids found? So, there are circular plasmids in comparison with other DNA strands, and plasmids are found in bacteria, archaea, and eukaryotic organisms. 4. What is the DNA sequence that marks the end of transcription? What is the term of it? What marks the beginning? So, the end is a terminator, and the beginning is a promoter. And what is an industrial use for genetic engineering? It's biofuels. Yeah. Thank you all for coming to see this video on genetically modified organisms and gene editing. Thank you so much. And I'm Hans, and I'll see you next time. Bye.